Okay, good morning class. Welcome to class today. Today I want us to look at carbohydrate metabolism. And metabolism is a biochemical reaction in which the reaction occurs in the living cell of living organisms. Now, living organism or each cell of living organism require energy, and this energy comes from the breakdown or metabolism of carbohydrates. Now we have 5 pathways through which the energy is formed and is enabled to be formed by the cells and used by the cells for their normal functioning. Number 1 is glycolysis. Glycolysis basically involves the conversion of glucose to pyruvate. Then number 2 is gluconeogenesis. There is the conversion of the pyruvate to glucose. Then we have number 3 is glycogenesis. That is the conversion of glucose to glycogen. And then also number 4 we have glycogenolysis, the breakdown of glycogen to form glucose. The levels of the glucose are very high in the blood. And then we have the Krebs cycle. Now this is a continuation of the glycolysis in which the pyruvate is converted to acetyl-CoA, then eventually to form the energy and CO2. Now, I want us to look at the first pathway, that is glycolysis. Now, glycolysis occurs in 2 phases. We have a preparatory phase, and also we have the payoff phase. And I want us to begin with the preparatory phase of glycolysis. Now, in preparatory phase, the glucose molecule is converted to glucose 6-phosphate. Okay. So here we have ATP being reduced to ADP. In the second step, maybe you can indicate in the diagram, that is a glucose molecule. So glucose converted to glucose 6-phosphate, which will be this way. This will be our glucose 6-phosphate. This OH. So this is where we have our phosphate attaching. So we form the glucose 6-phosphate. The enzyme involved in this first step of preparatory phase is hexokinase. The hexokinase enzyme. Now in step number 2, glucose 6-phosphate is isomerized by the enzyme isomerase to form fructose 6-phosphate. The enzyme is called isomerase enzyme. That is the enzyme. Now, in this particular reaction, the main difference we have here is that instead of we are changing the isomer, rather we are having a different isomer, that is from the normal glucose to fructose. That is, we are going to have a structure like this one. So our phosphate is attached in carbon number 6. That is fructose 6-phosphate. Now in step number 3, the fructose 6-phosphate is phosphorylated. It undergoes phosphorylation reaction, in which again we have ATP involved or reduced to form ATP, and the enzyme is called phosphofructokinase 1. Fructokinase 1. This forms fructose 1,6-bisphosphate. So basically, we are adding an additional phosphate to fructose 6-phosphate. When we do so, we are going to have an isomer or a molecule of this type. So our molecule also that's called that kind. So we have here the first phosphate in carbon number 1. This is carbon number 1. That's why we call it fructose 1, meaning that it's a phosphate in carbon number 1 and carbon number 6. So carbon number 1 phosphate is there, and then this other one, the phosphate, is in this particular carbon atom, number 6. This is where we have our phosphate molecule. So we form fructose 1,6-bisphosphate. Now in step number 4 of the preparatory phase of glycolysis, we have the fructose 1,6-bisphosphate being cleaved. So we have fructose 1,6-bisphosphate cleaved, cleaved to form dihydroxyacetone. Dihydroxyacetone is a 3-carbon sugar, and also we form another one, glyceraldehyde 3-phosphate. 3-phosphate. The enzyme involved is called aldolase. Aldolase enzyme. So this is step number 4. We form 2 molecules, a 3-carbon sugar. Remember, this is a 6-carbon sugar, which is called fructose 1,6-bisphosphate, being cleaved or broken down to form dihydroxyacetone phosphate, 3-phosphate, and glyceraldehyde 3-phosphate. This is how our dihydroxyacetone phosphate molecule look like. 3-phosphate. So we have it there. The 3-carbon sugar. And this one is called dihydroxyacetone phosphate, and also we form glyceraldehyde 3-phosphate, another 3-carbon sugar, which is an aldose. This is a ketose, and this is an aldose sugar. Okay. So we have our phosphate in carbon number 3. So we call it 3-phosphate. 1, 2, 3. Phosphate in carbon number 3. Phosphate in carbon number 3. 3-phosphate. Okay. The enzyme we have said involved in particular cleavage reaction is called aldolase enzyme. Now, Okay, now we can convert this one further to form that, and so we extend this particular reaction. So we go to step number 5, in which glyceraldehyde 3-phosphate is converted to glyceraldehyde. It's converted to 1,3-bisphosphoglycerate. Bisphosphoglycerate. Okay? That is a 3-carbon sugar which is of this kind. So, we have the phosphate in carbon number, let's say there, and then the other phosphate. So we have 2 phosphate. The other one we add there. So that is how we form the 1,3-bisphosphoglycerate, and the enzyme is called bisphosphoglycerate, glycerate dehydrogenase enzyme. Thank you.